So just thanks to Jay and Wolfram and the organisers for giving us an opportunity to, one, leave Australia. So this is the second time I've left Australia in the last 3 years, and to come and participate in this very exciting meeting. It's been really enjoyable so far, and I think today's sessions look really good as well. So I'm going to talk to you a little bit about the work that we've been doing in the lab, and when I say we, I mean Evangeline and Nada, who've done the majority of this work. And we've really, much like Mauro Jacques was saying yesterday, we're really using transcription as analysis as a way to look for pathways, to look for novel avenues for treating disease. So I don't know if you all know, but I live in Melbourne, and Melbourne was the longest lockdown city globally, and so we had a lot of time to develop new hobbies. Like some people did baking sourdough, some people took up beekeeping, and I thought I'd really work on my graphic illustrations. So this is a really anatomically correct heart from an embryo, and what we're really interested in is how do we turn that into the sort of beast that sits inside your chest that beats for 2 billion times, and we're interested in sort of fetal development, and more increasingly with Enzo Perello, we're really interested in this concept of maturation postnatally, and the sort of metabolic switches and changes that go on in the transcriptional landscape as post-birth. So our sort of start point, and I just want to point out the sort of utility of biobanks. We really relied on Sydney Heart Bank for this, and we've established a Melbourne Children's Tissue Heart Bank, and these have been really powerful research enablers for us to try and collect these sort of rare human samples with fetal, neonatal, child, adolescent and adult and heart tissue. And what Evangeline did was try and work out ways to sort of isolate single nuclei from these left ventricular portions. To essentially dissect them, break them up mechanically, some homogenisation, centrifuge, and then isolate nuclei, and use the 10x platform for RNA-seq. And in parallel, we also used PCM1 to isolate cardiomyocyte nuclei by flow cytometry, and we use that for bulk RNA-seq and importantly for ATAC-seq, which gave us an avenue into what was happening at a sort of chromosome level. And so when we look at this profile, you can see the hearts really have the same collection of cell types, regardless of fetal, young and adult, which was reassuring, I guess. And one of the things that we observed by sort of this sort of GO analysis was there was really all the cells in the heart undertook a transcriptional maturation during development and during aging. It was just sort of highlighted the cardiomyocytes. So the things that went up were, TCA cycles, classic electron transporters. Things that went down were cell cycle regulators, which, as we know, the heart's sort of post-mitotic after birth. So, but the same is true for every cell type that we identified. So all of them are getting cues throughout development to drive maturation. And so, this single nuclear RNA-seq captured this spectrum of changes across from fetal to late adulthood. And so one of the sort of key findings that Evangeline identified was sort of this age-dependent remodelling of chromatin. And so we also used mouse models as well as the human. And this is converted from Macintosh to Microsoft's Office here. Anyway, so this is ATAC-seq and RNA-seq, and as you would expect, as the chromatin opens up, you get a higher expression, so you sort of expect a correlation. It's not so strict in the mouse, but the GO terms that are sort of identified with the same sort of things like cell cycle, chromosome division, and then what we were able to do then is do this sort of gene set analysis and look for things that were variable across these genes. And so the classic sort of cell cycle regulators were up in the fetal samples and getting lower in the adult. And one thing that sort of piqued our interest, given the sort of differences in cardiac disease in male and female and the differences in sort of the biology, was we identified the sort of sex steroid receptors as a cluster that was sort of enriched in adulthood. So the hypothesis then was perhaps these sex steroids were playing a role in maturation of cardiomyocyte muscle. So this is, some of the data. So this is expression of the progesterone receptor, and you can see it's higher in both male and female adult compared to fetal. This is a tax-seq data. It's a bit hard to see on the colours, but essentially the open accessible regions were much higher in adult in the purple and young in the orange than it was in fetal or in IPS-derived cardiomyocytes. So IPS-derived cardiomyocytes tend to get to mid-fetal stage. So I just realised that I'd prepared this talk and I didn't have a single movie, so I just put this in so I didn't get chucked out of the cardiovascular bioengineering club. So this is the system we use. This is developed by Richard Mills in James Hudson and Isabel Perello's lab, and we use this to have a look and see whether we could add progesterone to improve the functionality. And so the experimental design was to set up the cardiac organoids and then treat from day 20 for 4 days and collect. And by filming the movement of the poles, we can interpret muscle force and electrical kinetics, activation and beat rate. So when you look at each dot is a single organoid. So when we added vehicle or testosterone, there wasn't much difference. When we added progesterone, there's a little bit of increase in force that made statistical cutoff. When we added a progesterone inhibitor, that blocked that, and the same was true with contractile rate. They started to slow down, so that's what you would expect. These organoids are fetal-like, so they beat a little bit faster than an adult bit of heart. So we were, we said it doesn't obviously mature them completely. Obviously, there's all sorts of cues that are going on: stretch, biophysical cues, hormonal cues, and the actual work that a heart has to do to pump blood. They all contribute to maturation, but at least there's a component with this progesterone pathway that helps promote it, not only in vitro. I don't have the time to show the data, but also in the mouse model as well by transfection of adenovirus. So I think our take homes were that this single cell and a tax-seq together had quite a lot of utility in identifying pathways that might be modifiable. Definitely the sex stories play a role in sort of heart development maturation, and we're currently pursuing that in the lab to try and dissect that further. And we feel that progesterone treatment may promote maturation in vivo. So I'll just change gears now and tell you a little bit about what Nate has been doing. Again, here's my highly anatomically correct heart. We had a disease setting, and then what often happens in disease and is reported is this activation of what's called a fetal gene program. And that's proposed to be protective of the heart, so it changes the sort of metabolism to a more fetal-like state, changes some of the sarcomeric proteins back to those that are expressed early in development. And we felt, based on what we'd observed in the data I've just shown you from Evangeline, that perhaps we could use a similar approach to target the transcriptional networks that actually, and sort of, in a sense, define this fetal gene program in a disease setting. So the experimental program was the same. Again, we made use of the tissue banks that we've built up. And so we had 3 fetal samples, some non-diseased children, and dilated cardiomyopathy examples. So the dilated cardiomyopathy was all genetic. So we'd sequenced them, and we knew that there were 2 of the children carried mutations in desmoplakin, 1 in cardiac alpha-actinin, and the other child had a double mutation, 1 in MYH6 and 1 in cardiac troponin C1. So this is, in a sense, a genetic model of dilated cardiomyopathy with various genes perturbed. So again, this is the single cell data. So again, we get the same collection of cell types. There's no real striking difference between sort of early childhood, non-diseased. The only thing that really is apparent is this collection of proliferative cardiomyocytes that are lost after birth. So these children are at least 5 years old. And then really no major striking difference in cell composition in the diseased heart either, which we were probably expecting some differences, but didn't really reach statistical significance. If we graph it out this way, cardiomyocytes in purple. In the DCM, you can see there's probably more endothelial type cells and some fibroblasts, but it doesn't really reach statistical power with the three tissues that we had. So we used this to do the single cell sequencing and then identify genes that were differentially regulated, and we didn't actually find that many. About 8% were genes that were fetal or developmentally regulated genes that we could identify. So 74 that were up, so this is fetal versus normal, and then disease versus normal, the intersection was 74, and then 89 that were down. So when we have a look at the sort of GO terms, these in a sense fit the hypothesis that they're reactivating the early cardiomyogenic program, so muscle organ development, cell adhesion, morphogenesis, muscle contractility. The downregulated ones were things that were a bit different, so vesicle mediated innate immune response was down, cell motility was also down. We then said, well, that there'll be different genes, different cell types within the section of ventricle that we took, whether it's strong outer curvature muscle or muscle that's closer into the trabeculae, but there wasn't really that sort of difference between any of the three, and these are sort of the cardiomyocyte lineages that we could detect, and the changes that we observed aren't restricted to a given lineage. So it's not the emergence of a new cell type, it's globally dysregulated across the myocardium. And the other interesting finding, I guess, was we found fibroblast genes were also reactivating a sort of fetal gene program. So, and a lot of them look to be sort of this sort of myofibroblast type phenotype. And interestingly, one of the 57 genes is MEOX1, which may be the driver of this adoption of the phenotype in these fibroblasts. So just as an advertisement, we're building a website that will be freely available called Heart Explorer. And this is what it looks like. I've just pulled out MEX1. So this is the UMAP sequences, and you can see down here, there's your fetal sample and your dilated cardiomyopathy sample and your non-disease sample. So I think this is live. We haven't published yet. We're in revision. We're hopeful. But I think it's live, so if you want to go and have a look at your favourite genes and your favourite gene set, feel free. What we hope to do is build on this and add further data sets. Stephanie Dimler's lab has another data set from DCM. She spoke yesterday, which was in circulation, is another nice data set. So we think this will be a good tool for the community to use, and we're hoping to extract some money to pay for someone to develop it further so that we can utilise it. You can also use it for pulling out your gene sets in cardiomyocytes and looking for your favourite gene sets and your areas of interest, whether it's endothelial, fibroblasts or whatever. So there's all drop-down menus, and it is live, so feel free to go and have a look. That's in the wrong spot, so we'll just go over that. I think what the take-home from it, I guess, is that only a small subset of the fetal developmental regulated genes are activated. We didn't see fetal-like proliferative cardiomyocytes in the disease setting. There's also fibroblasts that are upregulated, and I think one of the things we're sort of considering is this is just a dilated cardiomyopathy, genetically driven cardiomyopathy. In infarcts and in pressure overload and in other models, you also get this activation. So it remains to be determined whether the same response is seen in those different disease settings or whether this is always seen. So I'll thank there. I really would like to thank our patients and families. It's very humbling. They always give generously of their time at a really stressful time for patients and their families. The Melbourne Heart Tissue Bank is run by Adam Pears. Collaborate with Enzo Perello, so we sort of jointly run a group now and collaborate closely with James Hudson and the Sydney Heart Bank's Christos Remedios, and these are our funders. This is the lab, and so this is our lab retreat, and the reason they're all smiling is that they got to go to the beach and Enzo and I were stuck on a Zoom meeting. And we're looking for people. If you're young and you want to live in Australia and see koalas and kangaroos, please come and talk to me. And also, young Julian up there has a poster. So go and see his poster, please. Thanks very much.